So for the renal corpuscle anatomy, I'm going to use this model here and this model here. You can see a couple things on this model that you can't on this one, but this one's a lot easier to see. So we'll start off with our afferent and efferent arterioles. So this would be your afferent arteriole here, bringing the blood in, and then your efferent arteriole here, bringing the blood out. On your afferent arteriole, you're also going to find your juxtaglomerular cells. Okay? Your afferent arteriole and your ascending limb of the loop of Henle right here, these two make up the juxtaglomerular apparatus. So on here it would be these two. This is your juxtaglomerular cells, and these two make up the juxtaglomerular apparatus. So we have your glomerulus, which is just the network of capillaries inside of the Bowman's capsule. Now the Bowman's capsule has a parietal layer, which would be here, and then it has a visceral layer that lays on top of the glomerulus. Okay? Now, on the visceral layer, we have these cells called podocytes here, and we have the filtration slits. Now, when the filtrate is squeezed out of this vessel, it flows through the filtration slit and ends up in the Bowman's space. So the Bowman space is just any area in this capsule that is not occupied by the glomerulus. The pedicels of the podocyte, you can't see them on this model, but they would be the little legs sticking out of this podocyte. We also have the proximal convoluted tubule, which would be here and here. Now, the proximal convoluted tubule has microvilli, which help to increase the surface area and absorption that is happening here, reabsorption that's happening here. So if you can remember that, that'll kind of help you remember that this is the proximal convoluted tubule. We have the ascending limb of the loop of Henle, which is surrounded by the macula densa. So these green cells here would be macula densa. And that concludes everything that we need to know about the renal corpuscle anatomy.